You see it every single day, right? Splashed across your news feed, this new drug cures this amazing diet, prevents this. We're just constantly bombarded with these medical headlines. And look, it's exciting stuff for sure. But underneath all that noise, there's a really, really important question we should all be asking. I mean, this is the real question, isn't it? It's the core of everything. So a study says a drug works. Great. But how do we know if that result applies to the people we actually care about? Our family, our patients, or heck, even ourselves. Okay, so let's really dig into this. To really get our heads around that big question, it helps to break it down into two separate pieces. First, we're going to look inside the trial and ask a simple question. How did they even get that number? And then we'll step outside the trial and ask, okay, who does this number actually apply to? It's all about getting the result right and then making that result relevant. So let's tackle that first part. All right, first step. Let's go inside the clinical trial because, and this is key, it turns out that the first number you see in a headline, that simple, straightforward result, well, it's often not the full story. Now, you've probably heard of randomized controlled trials, right? The famous RCTs. They're called the gold standard for a reason. I mean, the whole idea is pretty genius. You randomly put people into different groups, and that helps you isolate whether a treatment actually works. But here's the kicker. Even with these top-tier trials, that initial unadjusted number you see, it can be, well, a little bit misleading. So why would researchers bother adjusting that first result? Well, there are some really compelling reasons. For one, it gives you a much better interpretation. You get a number that's way more specific to an actual patient. Second, and this is a big one, it gives you more statistical power. Think of it as getting more bang for your research buck. You get the same level of certainty with fewer people in the trial. And third, it lets you correct for little imbalances that happen just by the look of the draw. Like one group just happening to be a bit older than the other. Okay, so this brings us to a slightly tricky, but honestly, super interesting statistical concept called non-collapsibility. I know, it sounds a bit technical, but here's the core idea. When researchers adjust for something like a patient's age, they're not just fixing that imbalance we talked about. They're actually asking a totally different, more specific question. They're shifting from asking, what's the average effect for the whole group, to asking, what's the effect for an individual person of a certain age? And that little shift from the group view to the individual view can totally change the answer, often uncovering a much stronger effect. I know that might sound a little abstract, a bit in the weeds, so let's make this super concrete with a fantastic real-world example. We're going to look at a huge landmark heart attack study called the GUSTO-I trial. It's the perfect case study to see how all this adjustment stuff actually plays out. So in this trial, they took a bunch of heart attack patients and randomly gave them one of two different clot-busting drugs, one called TPA, the other called SK. Now, if you just look at the raw, unadjusted numbers, here's what you see. The TPA group had a 6.3% death rate, and the SK group had a 7.3% rate. Okay, so that's a 1 percentage point difference. This is exactly the kind of simple number you'd see in a news headline. Okay, now statisticians don't usually talk in percentages. They translate that 1 point difference into something called a regression coefficient. In this case, it's negative 0.159. Don't worry about the number itself. All you need to know is that because it's negative, it means the treatment was beneficial. It lowered the risk. So let's call this our baseline. This is the simple, unadjusted effect. Now, watch what happens next. Now, the researchers do one simple thing. They adjust for the patient's age, and just like that, the number changes to negative 0.188. Remember, a bigger negative number is better. It means a stronger beneficial effect. All of a sudden, the drug looks even more effective. In fact, the benefit just grew by about 18%. So the big flashing question is, why? Where did that extra benefit come from? And here is the breakdown. It's fascinating. That 18% jump in effectiveness wasn't from just one thing. It was almost a perfect 50-50 split. About half of it was from simply correcting that age imbalance, that little fluke of chance. But the other half, that came from our tricky friend, non-collapsibility. By asking that more precise, individual-level question, they literally uncovered a stronger treatment effect that was kind of hidden when you just looked at the simple group average. Pretty amazing, right? Okay, so we've just journeyed deep inside the trial to see how researchers can really sharpen their pencils and get a more accurate number. But that's only half the battle. Now we have to ask this second huge question. Even if we have this perfectly calculated, beautifully adjusted number, who does it actually apply to out here in the real world? And honestly, this can be a massive problem. The people who participate in a clinical trial, well, they often don't look like the diverse mix of patients we see in the real world. A really powerful and frankly concerning example comes from cancer research. We know that African Americans often have a higher risk for many types of cancer, but they are systematically, consistently underrepresented in the clinical trials that are testing the very drugs that could help them. This whole challenge of applying study results to the real world, it's called external validity. And it kind of comes in two main flavors. The first one is called generalizability. The easiest way to think about this is when your study group is just a small sample of a much larger group you care about. So you do a trial in, say, California. The question is, can you generalize those results to the entire country? The second flavor, transportability, is even trickier. This is when you want to take your results and, well, transport them to a completely different population. The study group isn't even a small part of the target group. The classic example is, can you take the results from a study done entirely in Japan and have any confidence they'll apply to patients in Brazil? It's a much bigger leap. So this is the big challenge, right? How do you build a bridge between the clean, controlled, almost sterile world of a clinical trial and the messy, complicated reality of an actual doctor's office? Well, the good news is researchers aren't just throwing their hands up. There are some really powerful statistical tools designed to do exactly this. And this quote from researchers Dikchar and Rose, I think it just perfectly sums up everything we've been talking about. What they're saying is, look, getting the number right inside the trial, what we call internal validity, that's absolutely essential. But it's not enough. It's only half the story. To get an answer that's actually useful in the real world, you have to consider external validity too. And thankfully, there's a pretty clear roadmap for how to do this. It's a 3-step process. Step 1, you assess dissimilarity. That's just a fancy way of saying you measure exactly how different your trial patients are from your real-world patients. Step 2, you evaluate for something called heterogeneity. This is where you ask, hey, does this drug work the same for everyone, or does it work better for men than women, for older folks than younger folks? And then step 3, if you find important differences, you correct for that bias. You use smart statistical methods to re-weight your trial results so they give you a better estimate for the actual population you're trying to treat. And that brings us right back to where we started. We've taken a journey deep inside a clinical trial to see how that headline number gets calculated and refined. And we've stepped outside to see the challenges of applying it to the real world. So after all that, what's the big takeaway? What should we all remember? It really just boils down to these two simple but incredibly powerful questions. The next time you see one of those breathless headlines about a new medical study, don't just stop at the headline. I want you to ask yourself two things. First, how did they get that number? Was it adjusted? Was it for the group or for the individual? And second, just as important, who does this number actually apply to? If you can ask those two questions, you'll be able to see beyond the hype, beyond the headline, and get to a much, much deeper and more useful understanding of how science really works.